So I think last time we left off, we were talking about roots, and today we're going to continue talking about the shoot system, which is the stuff above the ground. And remember, when we talk about growth, different parts of the plant have different terms used for that growth. So when we're talking about stems, and we're talking about the outside tissue, the tissue that gives rise to that's called the protoderm. So that's going to give rise to the epidermis. The procambium, anytime you see the word procambium, you're giving, you're building vascular tissue, xylem and phloem. And I think I mentioned this already, but procambium, when you talk about the xylem and phloem together, they call that the steel of the plant. I think we mentioned that when we talked about roots. Same thing applies with stems, and they come in different forms if it's a dicot or a monocot, just like it did when we talked about the roots. Stems do a similar thing where they're arranged differently, but it's kind of the opposite of each other, as I'll show you. So the xylem and phloem are in what are called vascular bundles. They're groups together, and in dicots they're found in a ring, and in monocots they're found scattered throughout. And in the dicots, there's going to be a center portion of non-vascular tissue called the pith. And you should remember that when we did monocots, the monocot root had a pith, but not the dicot root. Now it's the opposite, and we have the dicot stem has a pith, and the monocot stem does not. And then the stuff in the middle is called the ground meristem, and that gives rise to the cortex. This is what a vascular bundle looks like. Again, all of them together is called the steel of the plant. It's all the vascular tissue. And you often have these fibrous cells here called sclerenchyma, which is a really hard word to say, but it's fun. Anyway, so that's like a supportive tissue. And then this is really the vascular tissue. This whole thing's the bundle, but this is the phloem right here, the green tissue here. Remember, phloem transports food primarily. And then you have these sort of red colored rings here. Those are xylem, and those are usually xylem are used to transport xylem. No, xylem is used to transport water. Okay, and that happens in stems and leaves as well. So you might remember back when I showed you the dicot and monocot root in the last lecture, this is now a dicot stem and a monocot stem, and you want to be able to make sure you can tell the difference between monocot, dicot, root, and stem, and vice versa. Okay, so here's my vascular bundles. In my dicot stem, notice there's a pith right here in the center, whereas over here, there's no separation. The vascular bundles are scattered throughout. This is all sort of interconnected. There's no central area that is surrounded by vascular tissue, so there's no pith like there is in this one here. Okay, let's see, did I miss anything there? I got my ground tissue, I got my pith, and I got my cortex, right? The outer tissue outside of the vascular tissue. Okay, now the next thing, and this is very different than roots, is not only do they grow through apical meristematic tissue like we did with roots, but we also now have what's called secondary growth. This is getting wider or thicker. And that happens with at least two kinds of tissue. One is called the vascular cambium, and that produces secondary xylem and secondary phloem. And the other is called the cork cambium. Remember, cambium, whenever you see that word, we're making, it's like stem cells, and we're making something new. So the cork cambium is going to produce the cork and another tissue which is called the pheloderm, which is a very thin layer of parenchyma cells. And together, all those together make up what is called the periderm. So the periderm is the collection of cork cambium, cork, and pheloderm, which will make more sense when I show you it. Now, we have our tree or plant I like to think about trees because I think you're more familiar with how a tree grows in a sense. It's growing taller, obviously, like most plants do, and that's the apical meristematic tissue growing taller. But in addition to that, the tree is getting wider, and that's a different set of tissues. As you grow wider, you have a different set of stem cell-like tissues that are building that. So in secondary growth, what happens is the vascular cambium tissue, which is where the growth happens, will divide. It will go through mitosis and produce a new cell. And that new cell starts off as what we call a derivative cell. It is underived. It's sort of like an early stem cell kind of tissue. And then it's going to develop into like a kidney cell or a liver cell, but that's a plant, so it's not going to do that, obviously. But in this case, that derivative cell will turn into and become xylem. Okay? Then the vascular cambium again will go through mitosis and produce another cell, and it'll be another derivative cell, and that derivative cell then will become phloem. And what you should get from this here, as you see as this is growing, it's doing one side then the other. So what you end up with when you have your, let's say we go way over here over time, your vascular cambium is dividing and making new cells. And if you take the vascular cambium and go inward like this, that's all going to be secondary xylem. And if you go outward like that, that's going to be secondary phloem. Okay. So this is another view of that same thing. So if we take this as sort of, this shows you time, a graph over time, if you will, and then this shows you kind of the tree happening here. So if I take here's my growing tree, if I cut it, when you take a branch of a tree or a stem and you cut it, you've all seen the tree rings, and you can count how old a tree is based on those tree rings. This is how we're making those tree rings. Okay, so here's my vascular cambium right here, and the growth as this gets wider, you can see is occurring two ways. It's going this way and it's going that way. So does a tree grow from the outside in, or does it grow from the inside out? It's actually doing both. It's growing from that vascular cambium, and it's making new tissue pushing inward and outward. This is going to be the new secondary xylem, just like I showed you before. This is going to be the new secondary phloem. And then this is the cork cambium and all that stuff. That's the outer layer. If you pull a tree bark off, that's what you're getting there. Okay, so here's the same thing. You've probably seen this before. This is a tree that is 2 years old. Okay, so this is cut. This is where my vascular cambium is, right here. And then this is one ring, and that's another ring. This is secondary xylem. In both cases, this is my secondary phloem going that way. Okay, if I take a tree and I'll walk you through this, and then I'll show you it on a picture. But if you start at the very center of the tree and work your way outward, you would start off in the pith, then you would hit primary xylem, secondary xylem, vascular cambium, secondary phloem, primary phloem cortex, and then the periderm. And again, remember the periderm is those three layers: the phyloderm, cork cambium, and the cork. Again, I'll show you a picture of it because for me, the picture works better, but I show you it both ways. Okay. I've kind of hinted at this before, but I'll mention it here just so you know. When we talk about wood, I think when we talked about the cycads, the cycadophyta, first plants in the fossil record to show true secondary growth, botanists call wood, when you talk about wood, botanists call wood secondary xylem. things that have secondary xylem. That secondary xylem is the wood of the tree. So it's not all the stuff in the middle. It's not the bark on the outside. It's the secondary xylem. Okay. So here's a much, much older tree that has had a cross section cut where you're looking at it. This one might be, let's say, 80 years old or that kind of thing. And what you can see you have your bark here. And in this case, they're missing the cork or the cortex and the phyloderm. But here's my outside layers of bark. Here's my vascular cambium. And then here's my secondary phloem right there. So it's growing this way and it's growing that way. And what happens is you'll see in a tree, and a much older tree, the secondary xylem starts to take on sort of two different colors to it. And one's usually dark and one's usually lighter in color. In this picture, the outer layers are light and the inner layers are dark, but I've seen it the other way around too. But the point is they're different. And that secondary xylem, the deeper, older ones are called heartwood. And those tend to not transport water anymore. Those cells sort of die and they just stay there as support. And then the outermost ones are called the sapwood, and those still transport water. And you'll notice that the secondary phloem is actually very thin. And this is sort of deceiving because this looks like you have this big core in here, and this is often dead tissue. And so sometimes you'll have birds and things, drill a hole in here and nest on the inside. Well, as long as this vascular cambium is still in place, the inside part of the tree, often animals can live inside of that because that tissue is dead. And a tree can get really, really big and use a very thin layer of secondary xylem sapwood because it's much wider. So if you think about that, even though this is thicker right here from there to there, because this is a wider diameter, you can transport more water. Same thing with the food here. The secondary phloem is actually very kind of tiny, and it doesn't look like all that significant. You don't see much to it. But remember, it's on the outside layer. So the circumference of it is really, really big because it's in the outside layer of essentially like a circle. Okay. This is a nice picture that kind of shows you everything. Same kind of thing I showed you before, but here's my vascular cambium. Here's my secondary phloem. Here's my pith. And here's my primary xylem there. This is my secondary phloem. I'm not sure. I might have said that wrong. I'm going to try it again. I might have said something wrong in there. You know how I do that, where in my head I said it the right way, but then I heard myself say it. I'm not sure. So let's try that again, just in case. This is my vascular cambium right here, and it's going to give rise to my secondary phloem, which I see there. Okay. And my secondary xylem, which is right here. Okay. If I start in the center, this is the pith of the plant, and that first ring that I can see outside there, that is my primary xylem. Okay. And if I go all the way out, not to the cortex yet, but the outermost layer, so after all the secondary phloem, I can see on the outside the primary phloem. And then outside of that, I have my cortex layer, and then I have these three layers right there, my cork, cambium, my cork, and my phyloderm right there. And if you were to take a tree and pull what you call the bark off, this is what you're taking off right there. Okay. So the other thing, I'll point it out here, and then I'll show you it as I sort of zoom in. You'll notice that in these secondary xylem, see how some of these are big and then others are small. Okay, so here you have some big cells, and then over here you have some smaller ones. And that's because the secondary xylem in 1 year grows at a different rate. Early on, they call it spring wood or early wood. The growth occurs quickly, and there's usually a lot of water. and the cells grow faster and they grow bigger. They're not as strong, but they can transport more water. But as you go into summer and it starts to become drier, the cells tend to not grow as fast and it slows down and there's less water and they become denser and stronger but smaller in size. So if you look here, this right here is the summer wood, if you will, and this is the spring wood. And by comparing how many small ones you have versus how many big cells you have, you can kind of get a sense in 1 year how much water that tree got. And it differs by different trees, but that's sort of the nut of it there. Okay, and just like we had modified roots, we also have modified stems. So we have stolons that are horizontal stems that are above the ground, like you see there. We have rhizomes, which are the same thing, but they're underground. So strawberries reproduce in addition to flowers by stolons, but things like irises can reproduce using rhizomes, which are below the ground. Same idea, though, it's a stem. Tubers are swollen areas of a rhizome or a stolon. That's where potatoes come from. Remember I told you a sweet potato and a yam are a root, but a potato is not. A potato is a stem because a potato is a modified stem. It's a tuber, so it's different. Again, your friends probably aren't going to care about that, but that's just one of those things you got to know for Bio 2. You can tell your family, but that's how it goes. And then we have, we'll just lump these together. If you take botany, you get into more detail on this, but we call them bulbs and corms. They're basically a modified stem. Onions and garlic are example of that. Some of them have storage leaves with them and things of that sort. We'll just lump them together in that case, and that'll be good. So onions and garlic are different kinds of bulbs and corms as a modified stem. And then we have cactus pads, and cactus pads are called cladophylls. And so when you look at a cactus, the little spines on it are the spines. Those are actually the leaves that have been modified. And the cactus pad, which people eat, and I've actually had it. It tastes like jicama to me. It's pretty good, actually. Anyway, the cactus pad is actually a modified kind of photosynthetic stem. Okay. When you talk about leaves next, which are also part of the shoot system, there's a top to the leaf and there's a bottom to the leaf, and then there's the middle layers of the leaf. And mostly on the bottom of the leaves, plants have this, well, first you have the epidermis on both the top and the bottom, and on the underside there are little holes called stomata. And so a plant has these on the bottom of the leaf. That's actually surprisingly good for me, if I say so myself. Has these cells called guard cells, and the guard cells surround a hole. There's the hole, and the hole's called the stomata. And the guard cells can change shape, which open and close the stomata, and they allow things like CO2 to come in and water to leave and things like that. It's sort of a way that plants can do a type of breathing, if you will. Mainly plants need to get CO2 to do photosynthesis, and they do that by opening up the stomata. And the guard cells surround those stomata. And again, so the stomata is the opening, and the guard cells are the cells that are around it. And then some plants have a cuticle, which is a waxy coat on the outside. This can be thicker in some plants than others. And then you have the middle part of the leaf, which is called the mesophyll. And there's two parts to the mesophyll. One's called the palisade layer. That's used for photosynthesis. And then you have the spongy layer used for gas exchange. And just like we had modified stems, we also have modified leaves. So there are leaves that use small little, almost like little fingers that wrap around things, called tendrils. and tendrils are used for attachment. So plants that climb often, there are different ways that plants can attach to, say, a wall or a lattice or that kind of thing. Tendrils are one of those, and they're often modified leaves they use to do that. Bracts are modified leaves that surround a group of flowers, and that's what you're seeing here. So at Christmas time, my in-laws are really into the poinsettia plants, which have these big red flowers, they think, on them, but they're really not red. These are the flowers in here, little tiny yellow red flowers, and what you're seeing all on the red parts of it are actually the bracts. So those are modified leaves on it. And then there are spines, and I just mentioned that, like on a cactus there. So the leaves are modified for protection, like in spines. And then there are storage leaves that store water and excess nutrients like that. Succulent plants are big time storage leaf kinds of examples. They have these big thick leaves, and growing along the freeways often you'll see, I can't think of the name of it now, but anyway, as a kid we'd break them open and they're all juicy on the inside. And I'll think of the name of it. I'll have to put it up in the notes later on. I always forget this one. It's from Africa. Can't remember it. Anyway, it'll come to me. I always have trouble with that one. Okay. Now, the next part of what we're going to talk about today, and this will be the last thing we do on this particular lecture, is nutrients. So get a little bit more into different kinds of things that plants need. And we know a lot about what plants need by using these studies called hydroponic studies, which lots of people know about from growing all kinds of plants of all kinds of sorts. But the idea with hydroponics is you can grow plants without the soil, because you can replace the soil chemicals by adding these 17 essential elements that are needed by all plants. So this chart is one of those things you're going to have to know as well. You're going to need to know all 17 nutrients that a plant needs and what it's used for and about how much. Now, once again, like the geological time scale, this is that kind of thing that you first see it and your brain just goes, forget it, I'm dropping, I'm out of here. But it's not that bad. You're overthinking it. You need to break it down in a way that you can cope with it. And a lot of things are like that. So first of all, let's talk about macronutrients. Okay, macronutrients are things that plant needs in large amounts. Okay, plants need carbon. You know what carbon is, right? Where do you use carbon? Carbon is used in the phospholipid bilayers. If you look at the tails of the phospholipid bilayers in a cell wall, a plant membrane, any membrane, there's a whole bunch of carbon and hydrogen tails that make up that. Okay, we'll also use it in DNA and RNA, anything with a sugar in it, glucose. Carbon is sort of the backbone of all things. So that one should be easy for you. And it makes up a large part of the plant, 45%. And it's used all over the place. Okay, so that one's easy. Oxygen, hydrogen, okay, water, right? Hydrogen and oxygen, they make water. Lots of things are made out of water. And so you have hydrogen and oxygen as macronutrients in major organic compounds of all kinds of sorts, okay? So those first three should be easy, all right? Nitrogen. You ever heard of nitrogen before? Hopefully, right? So that one's a smaller percent, you see. But when you're talking about nucleic acids or proteins, you're talking about amino acids. They have that amino group. If you have an amino group, you have nitrogen in it. That one should be easy. Potassium, a little bit harder, maybe. Calcium for bones. You know, plants don't have bones, but but you're familiar with all of these right here. These are all ones you've heard of. Magnesium you've heard of, probably. Phosphorus, sulfur, chlorine. Now you're getting into the micronutrients. Iron. Now, there's some tricks though, and that's what you got to do is you got to go, okay, it's overwhelming at first, but the first 4 or 5 are easy out of 17. So that gets rid of a third of them. The next third are not that bad, but you have to notice like, okay, here's magnesium, which is a macronutrient, not bad, right? But here's manganese, which is a micronutrient. So that's one of those you got to pay attention to because they look the same. You could be fooled by that. So that one is one you got to go, okay, I'm going to pay attention to that. Boron, molybdenum, these are ones maybe you haven't heard of. Zinc, copper, and nickel, you've probably heard of those. Boron, maybe. So what you want to do is you want to focus on the ones you already know. You don't have to know the chemical formula. You do need to know if they're macronutrients or micronutrients. You don't need to memorize these numbers exactly. So if you say 45 and 45 and 6, and then all the others are 1% or less, that's fine for all those. 1% or less for the rest there of the macronutrients. And then these are all, the micronutrients are all 0.01% or less. Okay. And some of the functions are a little bit trickier and you're going to have to sort of pay attention to them. Like, for example, this one here, the molybdenum here, for nitrogen fixing bacteria. So some of those you've got to kind of focus on because their functions are kind of unique to plants. But many of them are, they're made nucleic acids and proteins and hormones. And a lot of the first sort of quarter of them are very simple or should be for you. So you ought to be able to handle that, I would think. Plants get those nutrients mainly from soil. And you can do hydroponic studies to learn how to get the nutrients without the soil, but normally the plants are growing in the soil and that's where they get their nutrients from. I'm going to stop here and I'm going to go over soil properties next time. Hope everyone is doing well and I'll see you soon.